internship, I was working with Tritium Biosciences on the transformation of bacteria with fluorescent proteins. So I did it at the Innova Bio here at Hagerstown Community College, and we worked with Tritium. And Dr. Mina, Dr. Kim, Dr. Dove, and Dr. Savita and we all worked together. So initially, what is a biofilm? So a biofilm is essentially just microbes living together, usually on a surface. So your teeth have a biofilm. Sometimes it can be seen, sometimes it can't be seen with the naked eye, so they use microscopes. But it also has an extracellular matrix, and that's kind of what makes it interesting. And also, in looking at biofilms, I found that different microbes can express different genes than their counterparts that aren't in a biofilm, which is interesting, and that's kind of what they want to work with. And also, biofilms are oftentimes in wounds, so they deal a lot with wound care. This is a slide that Dr. Kim sent me showing their setup, how they have the wells. So this does not have any growth of anything, it's just the media, whereas this one has this fungi. And it's a hydrogel that they use. And they have it in a 6-well little plate, and they can use that it fits under in their inverted fluorescence microscope, which we don't have.
No, we don't have one of those.
Okay, so the overall project was to transform different bacteria with plasmids, so that way we could look under a fluorescent microscope and fluorescence microscope. And what they want to do is they want to put different colored plasmids put one, like blue in one specific type of bacteria, so Pseudomonas, green in another, and yellow in another. They want to put them together in a biofilm and then be able to put them under the inverted fluorescence microscope to see how the different bacteria interact with each other. That's the overall goal. We decided to use electroporation to transform the bacteria with the plasmids. E. coli was really easy. Wasn't contaminated, was easy to work with, easy to grow, easy to transform. But the other bacteria that we used posed a lot of problems for us. So, Escherichia coli, gram-negative bacillus, we use DH5 alpha cells, easily and quickly grown, easy to transform. They grow faster than most other microbes, so don't really have to worry about contamination. The electroporation, so we used a BioRad electroporator, and we used the BioRad micropulsar protocol to make the electrocompetent cells and electroporate. Our results showed that the more piglo DNA, the more piglo DNA we used resulted in higher numbers of colonies. So, and then also this one we used 50 microliters of the cells with the DNA, the mixture, and this one we used 100 microliters, and it about doubled. Like this about doubled, if you can see the 100 nanograms versus the 200, it like doubled, and that was also more significant. These are the plates. This one over here is the 50 microliters with 100 nanograms of the piglo DNA, and this one is 50 microliters, of the 200 nanogram DNA. And that is 100 microliters, of the cells with 100 nanograms and 100 microliters with 200 nanograms of DNA.
Now these are cells that you made. You made electrically competent?
Yes.
You guys did? That's
yeah,
cool.
Those were electrocompetent cells that we spun down and we made and everything.
That's good skill.
Yeah, so we went to, Savit and I went to Tritium and we electroporated some E. coli with P-glow. And this is actually a picture that Dr. Hosan Kim took of the E. coli cells fluorescing under their fluorescent microscope. So the next one we used was Pseudomonas aeruginosa. Gram-negative bacillus. It's often found in wounds. It's really common in wounds, but it also poses an issue because it's antibiotic resistant, which I found. So in theory, it's a good candidate for this experiment because they want to deal with wound cultures. But it was ampicillin and kanamycin resistant. So Dr. Savit actually called Carolina, and they did confirm that the strain that we have, which is, I guess, an educational strain, it is ampicillin resistant. So I figured most would be ampicillin resistant. But most plasmids, they only code for ampicillin resistance. So we couldn't use it because it just grew a lawn. So we couldn't see which cells were transformed and which cells weren't, unless we used a UV light, which we did, but later on.
Okay.
So initially, there was contamination, which was our initial problem. So as you can see, there's like a flat type of colony. That's the Pseudomonas, but then there are these little dots that are kind of more white. That's contamination. So our actual plate was contaminated, so we couldn't figure out what was going on. The plating of the Pseudomonas, typically Pseudomonas has a green hue when you plate it, and I noticed that they weren't turning green. So that was cause for alarm. I was like, wait, I learned in microbiology that it turns green. It wasn't turning green, so we were concerned. So after much debate, we did a gram stain. And here you can see long strains. That's not what Pseudomonas is supposed to look like. So we did confirm that it was in fact contaminated.
So the stock culture that you got
So the actual plate
was contaminated.
From the time I got it.
Yeah. You never had a good culture
No.
to start with.
Don't know what it was
I
contaminated
know.
either. So I strict for isolation, and this is the contamination of it, which was what the cells looked like when we were getting them with electroporating. So it was all contamination that I was dealing with. So then we got a microquick culture from Carolina, and as you can see, this green hue, that's what Pseudomonas is supposed to look like, so I knew right off the bat it was Pseudomonas. So it was all consistent, it looked like Pseudomonas, it smelled like Pseudomonas, so we started working with that, made electrocompetent cells, electroporated it, but it grew a lawn. And it grew a lawn without any of the plasmid DNA, which means it was ampicillin resistant because it was on ampicillin plates. We increased the concentration of the ampicillin, still grew. And that's when Savita called Carolina, and they did confirm that it was supposed to be, in fact, ampicillin resistant, which made it kind of void for the purpose of our study. So next we used Staphylococcus aureus. Gram-positive cocci, also very common in wounds, very common in the medical setting. It's known for antibiotic resistance, MRSA. I think everyone has heard of that. So what we learned, actually, was that our transformations weren't successful. Followed the protocol in the BioRad manual, made the electrocompetent cells, followed everything as it was supposed to be done. Well, kind of. We didn't have B2 media, so then I made it, but it still didn't work. So then Savita found a paper online stating that there is a restriction barrier that inhibits the uptake of DNA. So, of foreign DNA. So what it does, it destroys the plasmid DNA. When it comes if it is transformed, it destroys it immediately. So I think there was a paper stating that we could use a specific strain of E. coli and grow the plasmids up in that, but we don't have that strain. So we did a gram stain. It's staph, which is indicative, like the circles, because it's cocci, and also how it's like clustered. That's indicative of staph. So when that didn't work, we moved on to bacillus. It is a gram-positive bacillus, which is a rod shape. I made the growth medium, the electrocompetent cells, the weakening agent, the electroporation buffer, followed the protocol. We didn't have some of the reagents. So I did try to kind of replace. There were some that I couldn't though. But I used tween 20. I used tween 20 instead of tween 80, so I messed that up on there. And I used sucrose instead of trehalose.
Yeah.
But it didn't work, unfortunately. There was no growth. These are the, what's the word I'm looking for?
Like the variables, I
Yeah,
guess.
thank you.
Yeah.
The variables that I used, we stuck with 40 microliters of cells, which actually in the protocol then I realized it said 60, but initially we did 40. It didn't work. So then there was a second attempt. So I tried this, 60 microliters of cells, I'm missing now, and we tried different variables. So we tried 80 nanograms and 800 nanograms, and we still did not see any transformed cells. There was no growth on any of the plates. So the reagents that we needed were ordered, but we didn't get them until I think last week, so I wasn't able to redo that. And the bacillus took a really long time to make the electrocompetent cells. I was here for
that day.
quite a long time working on that. So when that didn't work, we moved on to Klebsiella. It's gram-negative bacillus. It's a normal flora on human skin. It's common. We found it to be ampicillin and kanamycin resistant as well. And these are the problems that we kept coming up against us, basically, is everything's ampicillin resistant. And I think that's the problem. That's why Dr. Mina and Tritium, they want to work with wound cultures, because they want to use cold plasma and other sources to kill bacteria instead of antibiotics, because antibiotics are often ineffective. So Alon grew on the ampicillin plates, and then we actually, Dr. Dove increased the concentration of the ampicillin
Like
a lot.
Yeah.
It wasn't even Dove, it was like 5 times more concentrated, and it still grew. So, yeah. So we used 2 different methods. We used the cone method and the freeze-thaw method. In the paper, the freeze-thaw method was it was the better method to use, and we've used that in many of our biomanufacturing. We use that. You just freeze it and thaw it, and freeze it and thaw it. So then we purchased Topaz and M Orange plasmids. They came in stabs. And so what I did was I took some from the stab, streaked it on LB amp plates, and then what grew, I inoculated it into LB, and then I grew that in an overnight culture, and then I extracted the plasmids, and then I transformed the DH5 alpha cells, which is E. coli, with the extracted plasmids. So then I grew the transformed cells in LB, ampicillin, and arabinose. The arabinose is necessary for induction, to induce it. And then I created slides. So we weren't sure how to go about making slides. So I made a wet mount, I made 2 wet mounts, and I made 2 heat fixed slides, or 1 heat fixed slide, to view under the fluorescence microscope. The heat fixed slide, as you'll see, it shows more cells. The Topaz did fluoresce under the FIT-C filter, but the M Orange did not fluoresce under any of the filters we have, and that's the list of the filters that we have. We tried to do some researching, but we couldn't really find anything, so we were going to discuss with Dr. Lennon maybe what was going on there. That is the heat fix slide. As you can see, it is fluorescing and it is green. Topaz, I expected topaz to be blue. But it's more like on the yellow-green spectrum, so that's why this is green. And then this is the wet mount.
So you got it in there.
Mm-hmm. Yep. And then we ran a gel too.
Yeah, that's right, because it's there.
So that's on here too.
Cool.
And then this is the orange. So you can see the cells. They're there. But then this is under Texas red. But then when we turned the lights off, there was no fluorescence. So
I wonder if it quenches faster or something. I don't know. We'll have to figure it out.
I have no idea.
Somebody can figure it out next semester.
Okay, so we performed the gel of the orange and the topaz, and this is what the gel looks like. It broke, but still salvageable. Okay, so here is the topaz. Now, this is the ladder, labeled, and there were several trials. I added different concentrations of arabinose to the cells. Uninduced, there's no line here, or if there is, it's very, very faint. But as you move over into the higher concentrations of the arabinose, you can see that there's a line, a band there. So and that's at the 25 kb.
So it's getting turned on the more arabinose.
We think it's being induced.
We just can't see it.
Well, no, this is the topaz.
Oh, okay. It's not the orange.
So this is the orange.
Okay.
Which is the one that Seven and I were talking about earlier. I'm not really sure how to read it, but so here's the ladder, and this is where we were looking at, and this is the uninduced. But it kind of, this isn't a very good picture, but it kind of, we couldn't tell if there was a band here or not.
Right.
Basically, so we couldn't tell if it's actually being induced. So there would need to be more testing for that. So Dr. Savin and I went to Tritium to work on their clinical isolates because we couldn't actually bring them here. I plated 5 strains of Pseudomonas and 3 of the Klebsiella on amp plates to see if any were sensitive. And there was one strain that didn't grow, and that's the PA19660. But after we went down, we made electrocompetent cells. I electroporated and everything. Turns out it was ampicillin resistant. But we got pictures, so I electroporated cells with pGlow DNA and the PUC19. The PUC19 should not fluoresce, and it didn't. The pGlow, some did fluoresce, and it should have. We also electroporated some cells by themselves without DNA, and those were the ones that also grew on the ant plate. So that's, I don't know what I did when I was streaking, but apparently I did something wrong. So in the pictures that Dr. Kim sent, which are in the next few slides, there's some fluorescence under the UV light. So we think some were transformed. So here you can see that these look more green than those.
Right, they do.
And this has pGlow. And this is the PUC19 and no DNA.
So now
and this one is very green in comparison to that one. So I think the cells were transformed. We just couldn't
You can't
tell.
pick a cell.
Yeah.
You can't pick a colony.
So that's the problem with that in their isolates. But I mean, it was nice to see some fluorescence.
Yeah.
That was good. And this is under UV light for both of those. So, conclusion: the protocol we used for transformation for the E. coli cells, it was successful. It works. Every time we tried it, it works. But the other bacteria, not successful because of the various issues. So my suggestion is for the continuation of this project, clinical isolates that are ampicillin sensitive are necessary, because otherwise they're not going to be able to perform the experiments that they want.
Right, because all the fluorescent plasmids are AMP resistant. All
Yeah.
those fluorescent proteins
All the ones that we found were.
are in that AMP. Like
So-
if they were in something weird, like if they were in a different vector, right, then they might be more useful.
And that's why, because Elsie's was the thing that she was with, the plasmid that she was working
Yeah.
with, codes for the kanamycin
Yeah.
resistance. So that we did try the Pseudomonas and on the kanamycin, and
Yeah.
it was resistant to that too.
Yeah, so,
Which I remembered from microbiology. We used the
Yeah.
impregnated discs. Pseudomonas grew
right,
around most of them, but some of them it even grew over.
right.
So it's very resistant, which is a problem because it's common in wounds, and